Okay, so it's 12:02, and I think we'll go ahead and get started, and I'm sure people will trickle in while I'm speaking. So welcome everybody to our COVID-19 seminar series. And I just want to start with a brief announcement. So one of the goals of our working group is to connect researchers working on COVID-19 with resources on campus. And so we wanted to let everybody know that Anton Petrursky and Ariane Shirihai are willing to share 2 compatible human coronaviruses and their related reagents. So if you want to know more about this, you can email them, or you can email me and I can put you in touch with them. But they very kindly put out to us that they would like to get this out to the community. And I also want people to know that we're planning to develop a central repository for all these kinds of tools and reagents and things like that. So please stay tuned for more information, and we'll let you know once we have that all set up. And then now I'm delighted to introduce today's speaker, Pete Mullen. So Pete is an assistant project scientist in Dr. Heather Christofk's lab in the Department of Biological Chemistry here at UCLA. He earned his Master's of Biochemistry from the University of Oxford in England and a PhD from the University of Basel in Switzerland. And a little known fact is that Pete has lived in 5 different countries, but he thinks that LA is the best in terms of location and weather, and it's hard to argue with him on that score. His research focuses on how viruses alter the metabolism of host cells and how these changes can be exploited to develop therapeutic strategies against multiple different viruses, including, of course, now the SARS-CoV-2 virus. So my lab recently collaborated on a project with Pete, and I have to say he's an extremely talented scientist, and it's a real pleasure to work with him. So we are already looking forward to your talk, Pete, and the title of his talk is SARS-CoV-2 infection rewires host cell metabolism and is susceptible to MTOR inhibition. So Pete, please take it away.
Thank you for that introduction, Bridget, and also thank you, Bridget and Melody, for organising this amazing seminar series and inviting me and Heather to present our SARS-CoV-2 work. We're super excited to share this, and it's actually going to be the first time that we're going to share this work. And we couldn't think of a better place to present it, as Heather mentioned, and you did, about how UCLA is trying to get different groups and together with different skills and different reagents to be able to combat COVID-19. And this project, I think, is a great example of how that's been able to work, because it's involved maybe 5-6 different groups here. So our lab focuses on, one part of the lab focuses on viral metabolism. And the reason we focus on this is because viruses need materials generated by the host cell to replicate. They can't make their own nucleic acids or lipids or proteins. So they need to basically hijack the host cell. And about 1/3 of us in the Christofk lab is working on understanding how different viruses can alter metabolism of different host cells in different contexts. And we do this for 2 main reasons. The first reason is to uncover, we think that actually, because viruses have small genomes and have evolved to be, and therefore evolved to be efficient at hijacking host cell processes, we think they're a great system to uncover important mechanisms that control general metabolism. So if a virus can go in and maybe alter 1 step of a metabolic pathway to alter its whole output, we could maybe see if that step might be a very important step in that pathway. And that leads on to the second reason that we study viral metabolism. This might allow us to uncover targetable vulnerabilities that could be used to prevent and treat viral diseases, and maybe even other diseases such as cancer. So I'm just going to give a couple of examples of what our lab has done previously and published on viral metabolism with a couple of different viruses. And again, both these papers show the great collaborative spirit at UCLA. So the first one is by Min Tai, who was in the lab a few years ago. And this is on adenovirus. And I think this is just a striking visual so that you can see how viruses alter metabolism. So here on the left, we have 4 different mock plates at different time points post mock infection. And then in the middle, a wild type adenovirus. And you can just see the color of the media progressively gets more yellow. And because of the phenol red in the media, if it gets yellow, it means it's being acidified. So acid or lactate is probably being released from the cells into the media, causing this acidification. And this paper goes into looking at how viruses actually increase lactate secretion, which is actually showing that it's increasing glycolysis. Why the virus would need that, and it links it to how the virus actually specifically targets MYC, a common transcription factor, commonly activated in dysregulated in cancer. And if you get rid of the part of the virus that can interact with MYC, then you basically prevent the virus from replicating, and you also prevent the viral induced change in the metabolism. I think that's quite a striking image you can see there to see the effects. And another one recently, just last year, and this actually was a collaboration with Ren Sun and Vaiti Goswami, and Vaiti will be mentioned a lot in this talk, but this was our first collaboration with Vaiti. It was looking at Zika, and this is from a former grad student, Shivani, in the lab. And they, here I'm just showing a heat map of different metabolites under MTOR Zika virus infected cells. And here you can just see with the red, there's an increase in lots of different amino acids. So the virus is increasing amino acid production. And it does it specifically in human cells and not in mosquito cells. And you can, the paper's just looking at differences between the two different cells and why maybe the human cells are susceptible to death, whereas the mosquito cells aren't. So they were just two examples from our lab, but this is a review that our lab put out earlier this year. And you can see that there's been a lot of work focusing on lots of different viruses, RNA viruses, DNA viruses, encapsulated viruses. And this just summarizes some of that work, how putting different viruses into context of different metabolic pathways. So here you can see like Epstein Barr virus and hepatitis B virus can increase glucose uptake. Hepatitis C increases glutamine, adenovirus does. Here's a Zika affecting the TCA cycle. And so different viruses can affect metabolism in different ways. So it's not just a case that if we just know what one virus is doing to alter metabolism, all viruses will be the same. You really have to go into each virus individually, find out how it's affecting metabolism, which host proteins it may be affecting, so that you can then target it. So that leads to the questions we have for this project. So our first one is, like, how does SARS-CoV-2 infection alter host cell metabolism? And we can leverage our skill in our laboratory by using cutting-edge mass spec techniques to interrogate the metabolism. And then we also ask, okay, once we know this, how can we learn anything from that to target key metabolic regulators that might reduce SARS-CoV-2 replication, with the goal to maybe translating this into the clinic. I'll just say this is actually an image of the coronavirus. You can see the spike proteins here forming the crown, and it's taken from the NIH. So if we just focus on the first part first. So I'm just going to zoom in now on the TCA cycle. So the TCA cycle is in the mitochondria, and why we focused on this initially is because it actually, when people think of the mitochondria, you just think of ATP production and electron transport chain, and the TCA cycle does play a role in that, but it also leads to the production of many materials that the cell needs and also a virus would need to replicate. So you can see it can make amino acids, nucleotides, another way to make amino acids, lipids, sterols, all these materials that a virus would need to be able to package new viral particles and then get out of the cell. And there's two main routes into the TCA. There are other routes as well, but just for the purpose of this talk, I'm just going to focus on these two. There's two main carbon sources that feed into the TCA. One is from glucose and one is from glutamine. So a nice easy way to check what's going on in viral infected cells is to label cells with either with glucose or glutamine. So you can put on heavy labeled carbon on each of these metabolites, replace the normal glucose or glutamine in the media, leave it for a set amount of time, extract metabolites from the TCA, and then check if there's an increase in heavy labeled glucose, say in citrate, or heavy labeled glutamine in alpha-ketoglutarate. And then you can see if the virally infected cells are altering flow of these metabolites into the TCA. So to start with, I'm just going to show what happens when you replace all the glutamine in the media with heavy labeled glutamine during SARS-CoV-2 infection. And now I'd like to mention that all the infections were done by Vaiti and Gus in the Aruma Goswami lab. And I'm going to put in the bottom left basically every lab that's helped with this data. But you'll see Vaiti's lab is probably on most of the slides. So here I've chosen 5 metabolites from the TCA cycle. And in gray, you've got mock infected cells. and in red is the SARS-CoV-2 infected cells. And this is showing the amount of, so here if we look at citrate, so in mock infected cells, we see that about just under 0.8, just under 80% of the cell of the citrate is labeled by glutamine. That means that they've taken up this fully labeled glutamine, and the citrate in the TCA has incorporated, 80% of the citrate in the TCA has incorporated that labeled glutamine. And then the viral infected cells, it's gone down to below 60%. And we see this across the board. So these 5 metabolites in the TCA cycle, they seem to be being generated at a lower rate than in the virus cells and in the mock cells, which we thought was an interesting observation. So I'm now just going to go into this labeling pattern of glutamine in the TCA. So here you see glutamine has 5 carbons, and this glutamine can go into the TCA via glutamate to alpha ketoglutarate, still has 5 carbons, and it can cycle around. And you can tell based on how many carbons are labeled in each metabolite, how that glutamine has got to it. So there's 2 main routes in the TCA for glutamine to go around. So there's oxidative metabolism, which is the more traditional way you think of the TCA cycle. It's cycling in a nice clockwise rotation as we think of it in most diagrams. And you see that 5 carbons from glutamine end up getting like 4 carbons of fumarate, so fully labeled fumarate. By the time it gets found to citrate, there's still 4 carbons, but it gains a couple of extra carbons from the glucose up there. So we still have 4 labeled carbons for citrate and aconitrate, and this can keep cycling around. So basically, you can use this pattern to see, like, if you get 4 labeled carbons of citrate, you can presume that it's come from oxidative metabolism. But glutamine can also go in the reverse way, which is called reductive carboxylation. And this has been the focus of a lot of research in cancer cells. And reductive carboxylation can happen when the mitochondria might be slightly impaired, or when the cells need to create more lipids or aspartate. So glutamine in this case still goes in via glutamate to form 5 carbons of alpha ketoglutarate, and then it goes, instead of flowing in a clockwise direction to fumarate and malate, it goes the other way. So these 5 carbons go straight round to citrate. So now you have 5 carbons that are labeled from citrate instead of 4 from the oxidative, and it flows through to basically be 3 malates and 3 fumarates. Now, don't worry, you don't need to remember these. I will have them on the slides where I show data. I'll have these on here so you'll be able to follow it. But basically, to get from this, different metabolites have different labels from glutamine, and you can work out which direction the TCA is going in. And the idea would then be, if the TCA is flowing in one way, the other way might be impaired. So it might give us a hint of what the virus is doing to alter glutamine metabolism. So here, this is showing the data I showed on the other side, where I had the 5 metabolites and showing a reduction in glutamine labeling. This is now showing that, but with all the different types of labeling. So if we look at citrate again, you can see that if the colored is if it's got a glutamine label. So in the SARS-CoV-2 infected cells, you see there's fewer colors. That matches like last time. And then you can look into the individual colors. So the white means there's no label as last time. And if you look at say the green, this is an N+4 form. So there seems to be a lower amount of citrate that's labeled with 4 carbons from glutamine. And you can go all the way along. You can see again, this fumarate has lower M4, the malate has lower M4, and you can then so you can look at all the different labels. So what we noticed with that was, so if we go back to the oxidative arm, which I want to show you the labeling here, if we look at 4 metabolites, which we can use to basically see how oxidative metabolism is happening, so the N+4 fumarate and malate and citrate and aconitrate, we can see that actually these labels are reduced in the viral infected cells. So SARS-CoV-2 seems to be reducing the amount of label from oxidative metabolism through the TCA. And then if we look the other way with the reductive use, there's a slight reduction, but nowhere near the same amount. So it seems to be that SARS-CoV-2 is reducing oxidative metabolism, but maintaining or slightly reducing the use of reductive metabolism of glutamine, which is a little bit similar to what can happen in some cancers, and might be that, again, hinting that the virus is trying to maintain production of molecules that it needs to replicate, such as lipids and amino acids. So going into how the virus might be doing this, So this first step from alpha-ketoglutarate round to make succinyl-CoA and succinate involves an enzyme complex called alpha-ketoglutarate dehydrogenase. And this involves 3 different proteins. So it's a complex. These are the 3 the proteins, and they catalyze a reaction, much like a ubiquitin reaction where you have an E1, E2, and E3. And we thought, seeing as that was the first step from alpha-ketoglutarate in oxidative metabolism of glutamine, if we look at the levels of these proteins in the cells that are infected, that might give a hint as to what's going on with this reduction in oxidative metabolism. So this is in vero cells, and I should mention sorry, I didn't mention before that the metabolomics I've shown are in vero cells. So they're like a traditional cell that's used to study coronaviruses, but I will go into some more some actual human cell lines later. So vero cells are an African green monkey cell line, and they don't the antibodies we use don't pick up every protein. So the antibodies we use, don't work. Every antibody we use doesn't work in vero cell lines. So we're lucky to get something. So we were quite happy to find that we could determine the levels of 2 of these proteins in the complex. And you can look at OGDH. There's a striking reduction it's over 50% reduced in the vero cells in SARS-CoV-2 infected cells. And this is at the same time point as the metabolomics was done in. And then this other component, DLD, is also reduced, not as much, but still reduced. So it seems to be that the virus is reducing levels of this complex, which might explain why glutamine flow in the oxidative arm of the TCA is reduced as well. So then if we look the other way and we look at glucose entry, because it's not just glutamine that goes into the TCA, there can be glucose. So this is the glutamine that I showed previously. And if we look at glutamine, we see that it's the opposite. So the red is the virus. So we look at the metabolites, the same metabolites, and how much newly labeled carbon, how many carbons from glucose are being incorporated into these metabolites. And we see, as I mentioned, it's the opposite. Citrate, there's more citrate labeled with glucose. Same with alpha-ketoglutarate, fumarate, malate. So it seems to be from this data that the virus is switching, it's reducing the amount of glutamine that's coming into the TCA and increasing the amount of glucose. So we can do a similar thing where we look at how these metabolites are labeled as we did with the glutamine. So glucose incorporation is a little bit different. So glucose can be, at the end of glycolysis, can be split into 2 molecules of pyruvate. And then this pyruvate can enter the TCA cycle at 2 different points. And these different points lead to different labeling. So the first way is the way that most people think of pyruvate entering the TCA cycle. It's by generating acetyl-CoA via pyruvate dehydrogenase. And this means that 3 carbons from pyruvate just go to 2 carbons from acetyl-CoA. So if you pick up 2 carbon labeled, then it's probably coming from PDH. The other way is that pyruvate can be directly converted to oxaloacetate. And this maintains the 3 carbons from pyruvate to oxaloacetate. So then you'll have a 3 carbons labeled from pyruvate into the TCA. So we can look at this differential labeling again at the metabolites to see if we can determine which route that the virus is incorporating pyruvate and glucose, or is it just increasing them both? So again, I'm going to show the different colors represent the different amounts of carbon that are labeled. This is again in vero cells. Again, the infections were performed by Gus and Viti. And you see that with like alpha-ketoglutarate, we see an increase in this M5 form. That's probably a combination of the 2 pyruvate ways to bring pyruvate in. If you look at fumarate and malate, there's this increase in the yellow, which is the M3 form, which suggests that maybe the pyruvate carboxylase arm is being increased. So again, I'll just show this here to make it easy for people to follow. And so I'm just going to choose the malate here. If we look at malate, we can look at the different labeling pattern from glucose. So M0 means there's no label from this glucose here. So it's just unlabeled glucose. We see the reduction in that. So there's more labeled glucose, like I showed already. And then there's this increase in the M3 form, which suggests that glucose is being shunted through the pyruvate carboxylase arm rather than the pyruvate dehydrogenase arm to enter the TCA cycle. And this could be important because there's been numerous studies that show that increasing pyruvate carboxylase can maintain the cycling of the TCA cycle, when metabolites are being taken out. So if the virus is shunting off metabolites, say from citrate to create lipids and more acetyl-CoA and fatty acids and amino acids, then it needs to replenish this somehow. And if the glutamine arm has been shut down here a little bit by oxidative metabolism, then the virus can increase citrate levels by increasing oxaloacetate levels via pyruvate carboxylase. So we did the same. We decided to look at the two roots in pyruvate dehydrogenase and pyruvate carboxylase, the same as we did with the glutamine arm. And pyruvate dehydrogenase is very similar to alpha-ketoglutarate dehydrogenase. It actually shares one of its members is the same, DLD. These two are just different. So we have pyruvate dehydrogenase, DLAT, and DLD. And then we looked at the levels of these. So pyruvate carboxylase, we didn't see any change in levels. That doesn't mean its activity hasn't changed. And we're actually doing some experiments to determine whether its activity has changed. But level-wise, there was no difference with infected cells or non-infected cells. And then when we look at these members of the PGH complex, and this time we detected all three members, we could see again DLD is reduced in the SARS-CoV-2 infected cells. PDH is slightly reduced, and then DLAT, not as much as the others, but still slightly reduced. This number is the ratio of MOC to SARS-CoV-2 infected cells normalized to actin. So it seems to be that the pyruvate dehydrogenase complex is downregulated in SARS-CoV-2 infected cells, which might explain why there's more carbon entering through pyruvate carboxylase. And then, so these metabolites were all, these metabolomics were all performed in the Vero monkey cells. And so we wanted to look in a more relevant human system. And this is when Viti was already collaborating with Bridget on air liquid interface cultures from human stem cells or bronchial epithelial cells. And so we took advantage of that as well. We got in touch with Bridget, and she kindly agreed to let us use some of her air liquid interface cultures, which she's explained many times, I'm sure, to everyone here. But basically they can form a differentiated layer of lung cells where the bottom is exposed to media and the top's exposed to the air. So it mimics better the situation in a human. And then we can change the media underneath into our labeled media. And here you can see, this is just looking at glucose. So this is at 24 hours. It's an early time point in the NHBA and the ALI infection. And you can see the same things you saw in the Veros, where you see an increase in glucose label. So basically, the more color in the SARS-CoV-2 infected cells means more labeled glucose into the TCA in these 5 metabolites. And you can even see the yellows, the M3, which is the pyruvate carboxylase, that there seems to be a slight increase in that as well, in like aconitrate, citrate, and fumarate and malate. So this seems to match our Vero cells, which we're quite excited about. And then just thinking again about the glucose. So does pyruvate dehydrogenase, is it altered in other systems of infection? And Viti had collaborated on a cardiomyocyte infected, and this is published in Cell Reports Medicine. And they did RNA-seq on SARS-CoV-2 infected cardiomyocytes. And we mined that data and saw that there was a reduction in this blue in the pyruvate dehydrogenase complex members in SARS-CoV-2 infected cells. And there was also, in this system at least, an increase in pyruvate carboxylase. But this seems to match the expression changes and the metabolomics that we saw in our systems. And then also Viti, Bridget, and Catherine Platt's lab have been working on doing single-cell RNA-seq in SARS-CoV-2 infected air liquid interface cultures. And we've been working just to look at levels of these genes in SARS-CoV-2 infected cells. And quite a lot of these genes were lowly expressed in these cultures. But we did see, for example, in pyruvate dehydrogenase A1, we saw a slight reduction in the expression of that gene in SARS-CoV-2 infected cells. So SARS-CoV-2 seems to be having an effect on host cell metabolism. It seems to be altering carbon use in the TCA cycle, possibly to to increase production of materials that it needs to replicate. So now we want to know, can we target anything that would help reduce SARS-CoV-2 replication? Unfortunately, in the TCL cycle, there's not any approved drugs that can target that, apart from for mutant IDH inhibitors that are used in cancer. So we decided just to move out a little bit and see if there are any regulators and metabolic regulators that have FDA approved drugs that we could use that could inhibit this metabolic pathway. So this is where I'm going to bring in mTORC1. Now, mTORC1 is one of the key regulators of cell growth. This is a very simplified diagram of mTORC1. It basically signals through to increase synthesis of all the things that a virus would need: nucleotides, lipids, mRNA. It also increases glucose metabolism. And it's very tightly controlled in normal cells, but you have to have nutrients present, and you also have stimulation from growth factors. So if mTORC1 will only be signaled through for the cells to grow if you have these different signals coming through. And before the pandemic hit, I was studying actually how mTORC1 is dysregulated in adenovirus. And this is just some data from pre-pandemic times, which show that adenovirus increases mTORC1 activity. You can see this increase in phospho-S6K. And then when you take one of these nutrients away, for example, methionine, in mock-infected cells, you reduce mTORC1 signaling. Yet in the adenovirus infected cells, the virus can maintain signaling even when there's no nutrient there. And we show this with different amino acid depletions. So it seems to be at least adenovirus can maintain mTORC1 activity regardless of whether the nutrient's there. And so we decided that, seeing as mTORC1, it controls a lot of metabolism, and we have evidence in different viruses that mTORC1 is activated upon infection. And there are also clinically approved mTORC1 inhibitors that we could easily translate into the clinic. We thought this was a great target to see if viral replication can be inhibited with these. So we first went back to Vero cells, the workhorse of SARS-CoV-2 research up until now, and we saw an increase. So here, when you add SARS-CoV-2, we saw an increase in phospho-S6, which shows that mTORC1 is activated. We then moved on to two different patient ALI cultures. So these are not SARS-CoV-2 patients, just for different donors. And you see in both of these, we see an increase in phospho-S6. We see an increase in phospho-4-BP1. These are downstream targets of mTOR. So again, in these primary human cultures, we see an increase in mTORC1 activity, so that gave us great confidence that SARS-CoV-2, much like adenovirus, can increase mTORC1 activity. And then there was a question of whether SARS-CoV-2 if it's increasing mTORC1 activity through growth factors or nutrients. And there was some recent work that came out that showed that SARS-CoV-2 actually can increase ERK phosphorylation and PI3K activity. With some, they performed some proteomics and phosphoproteomics and saw that. So we decided to check in our ALI cultures again. So this is again with Viti and Bridget. And so we checked for these upstream growth factor components, and we saw even at 24 hours, this is when the metabolomics was done, we saw an increase in phospho-S6, as we saw before, phospho-4-BP1, which is downstream of mTORC1. Then upstream, these growth factors were also activated. You see an increase in phospho-ERK, slight increase in phospho-AKT, and you see this still at 48 hours. So it seems to be that the virus can activate mTORC1, maybe by increasing this growth factor signaling arm. So that gave us confidence then, and we've seen in multiple cell lines that mTORC1 is activated upon SARS-CoV-2 replication. And we also have a hint at how it might be increasing activation. And then when we were chatting with Viti about this, it turned out that Viti has been collaborating with Robert Demerceau at doing a screen in Vero cells to see for kinase inhibitors that might reduce SARS-CoV-2 replication. And it turned out that a lot of their hits were mTOR1, or mTOR inhibitors, I should say. And so this is some data from them, where these, I should say that bistuximab and taurin-2 are not FDA approved. They're just tool compounds. But if you look at this IF, where red is staining for double-stranded RNA, so that would be the virus. If you look when you add SARS-CoV-2 and a solvent, you see an increase in this red staining, which shows that there's virus present. And when you add these two different mTOR inhibitors, you see a reduction in the red, suggesting that inhibiting mTOR could reduce viral replication. So we then decided to move into FDA. So we took that data and was like, well, let's use some FDA approved mTOR inhibitors. There are currently 3 of them available: rapamycin, which is known as sirolimus as well, everolimus, and temsirolimus. And we did the same experiment. We just chose 1 dose of these inhibitors, added them onto infected cells, and this is showing a qPCR of SARS-CoV-2 gene. And you see in the red, which is the mock infected, compared to the gray, which is, so the red is the infected cells. You see an increase in the viral gene expression from the mock. And the gray bars are those SARS-CoV-2 treated cells, SARS-CoV-2 infected cells treated with the 3 different inhibitors. And we see varying degrees of decrease in gene expression, suggesting that maybe these drugs are having an effect in the viral cells. And just to show in these cells that mTOR is being inhibited, so on the left is mock infected, the right 4 are SARS-CoV-2 infected. You can see that nice increase in mTOR1 activity is shown by an increase in phosphorus 6. And then when you add the 3 different inhibitors, you decrease that. So again, we wanted to move from the viral cells into a more relevant line. So we went back to the air-liquid interface cultures with Bridget and Viti. And this is showing, similar to last time, this time it's showing a viral protein. So in the mock cells, we don't see any virus there. The green is showing the protein. So in SARS-CoV-2 infected cells, we see an increase in staining. And then when you add the drug, staining is hugely decreased. So it seems to be that in the ALI cells, rapamycin can decrease SARS-CoV-2 replication. If we look at another marker, the qPCR again of this viral gene, there's no expression in the mock cells. We see an increase, we see expression of the SARS-CoV-2 gene in the SARS-CoV-2 infected cells. And then this is hugely decreased when we add rapamycin, confirming these results. We also looked at a pan-SARS-CoV-2, using a pan-SARS-CoV-2 antibody. So this will detect lots of different proteins in SARS-CoV-2. So if you look at the third lane is the SARS-CoV-2 plus the rapamycin, you see a decrease in these SARS-CoV-2 proteins. This is just 2 different exposures. So in a few different ways, it seems to be that rapamycin can really decrease replication of SARS-CoV-2 in these air-liquid interface cultures. So I'm just going to summarize now. So for the first point, how does SARS-CoV-2 infection alter host cell metabolism? We've shown that reduced TCA cycle glutamine oxidation occurs in infected cells, and this might be compensated by increased pyruvate entry into the TCA cycle using pyruvate carboxylase in those SARS-CoV-2 infected cells. And the virus is probably doing that to maintain synthesis of the materials it needs to replicate. And then we took this knowledge to see if we can target these pathways using mTOR inhibitors. And we've shown that mTOR1 is activated in multiple systems of SARS-CoV-2 infection, and that when we use an FDA-approved inhibitor of mTOR1, rapamycin, we can reduce SARS-CoV-2 replication in multiple systems. So we have some next questions that we would like to pose, with the idea to try and translate this into the clinic. So one of the major things that we need to address is mTOR1 activated in COVID-19 patients? And we're actually working with UCLA Health and Sam French and Elaine Reed and PRP and TPCL and lots of people to actually interrogate those in a couple of different systems. We've been given access to COVID-19 patient autopsy lungs and also non-COVID-19 autopsy lungs. And we're staining them with the Shackelford lab with Melissa in there to determine whether mTOR1 is activated in those lungs. We've also working with the Plath lab on some samples from Elaine Reed on some patient aspirates to determine, into COVID-19 patient aspirates, to determine whether gene expression is altered in the cells that are infected or not in those aspirates. So that should help us explain if MTOR1 is actually activated in patients. And then we have to think, like, with what else would we need to translate this observation that MTOR1 inhibitors reduce SARS-CoV-2 replication into the clinic. We'd like to move forward into a mouse model of SARS-CoV-2 infection. Although I do think, again, whether that is possible or not will depend on whether we have BSL-3 access in the vivarium. So I think DLAM is working on getting that set up for VITE. So that's the end of the talk. There's lots of people I want to thank who contributed to this. As I mentioned at the start, it's been a hugely collaborative affair. VITE's lab, VITE and Gus, were amazing. They do all the infections. They go in any time of the day or night to infect cells, depending on our strange time points in the BSL-3 level lab. And without them, it would just be, this project just wouldn't have happened. Obviously, Heather, the Christofk lab, my mentor, and the first two on here, Nedas and Ernst, perform the metabolomics and analyze it. So without them, we wouldn't be able to do the metabolomics, and everyone else in the lab as well. Bridget, for providing these AI cultures, which add such relevance to this work and give us more confidence. So especially Runima and Shandani, who culture these cells and help treat the cells as well. Catherine Plath and Justin and William in the Plath lab will be performing all the patient aspirate analysis. The Shackford lab with Melissa will be helping with all the staining for the patient autopsies. And Robert at the MSSR for doing the screening with VITE, which show the mTOR inhibitors. PRP, TPCL, Sam French, Elaine Reed, and UCLA Health for being amazing at getting us access to patient samples and for getting us those samples in a timely and amazing way. And with that, I'd like to thank everyone for listening. And if anyone has any questions, I'd be happy to answer them.
Thanks so much, Pete. That's really exciting work, and hopefully it will translate to the clinic and sooner rather than later.
Yeah, hopefully.
That'd be great.
Well, we think it might be useful just as part of a cocktail of drugs. And what we're quite excited about with MTOR is that it does seem to have these viral effects in cells, but also it does have immunosuppressive effects, which you would think, oh, that might be bad in a viral disease, but especially for late stage, like severe COVID-19 patients, it is the immune response that kills them with the cytokine storm. So this might be a great way to introduce an immunosuppressant that's also an antiviral, which, so yeah, we think it could have great promise.
That's wonderful. Are there, if anybody has any questions, you can feel free to put them in the chat or to raise your hand or turn on your camera. That would be great. Maybe I can start with a question.
Yeah.
I always think about the fact that when you're doing your metabolomics or you're doing your Westerns, you're using all the cells, right? So some of them are infected and some of them are not infected. And I think a lot about this with our liquid interface cultures, because we already only get about 5% of the cells that are infected baseline. So I'm wondering if you're thinking that there's sort of like a bystander effect, or whether the cells next to the infected cells, the metabolism is changed in a different way, and whether what you're seeing is sort of the aggregate, or what you think about that, and whether you think the single cell sequencing approaches might be helpful for that.
Yeah, that's a great point, especially like with the vero cells, we do see high infection. So I think what we see there is probably what's happening in the cells. I think most are infected, but with the ALI, with the air liquid interface, yeah, like there's only 5-10% infected. And it could be that that's enough to be able to enable us to see this labeling change. But as you mentioned, I think it's more likely that factors are being secreted. Now, they could be metabolites, or they could be growth factors, or different signaling molecules that are affecting the other cells, maybe priming them for infection or not. It would be great to do single cell metabolomics. I don't think we're there yet, but there are some labs that are developing some cool technologies to actually do that. That would be great to answer, but I think for the timeliness, that would be really difficult. But yeah, it would be interesting. That's what we're wanting to do with a single cell RNA-seq. If we can actually look at those cells that are infected, even in the same dish compared to those that aren't, do we see changes in metabolic genes? It could be that we don't, for what you said, that because there's also having bystander effects, but that would be really interesting to see.
Cool, that sounds great. I'm wondering if there are any more questions. Oh, there we go, Melody.
I'll go next, Bridget.
Hey, Melody.
Hi, Pete. That was a really nice presentation and really nice data.
Thank you.
So I guess I was just wondering about the mTOR pathway, since as you described, I mean, it affects so many different processes, and I think it's also linked to aging. I'm just curious, what do you think in sort of the more susceptible, older sort of populations, right? If activation of the pathway by the virus could be more detrimental, and that if you guys are planning to look at the pathway in sort of from samples of different age also, and perhaps also other diseases like metabolism diseases that make them more predisposed.
Yeah, there's a lot of very interesting questions there that we're thinking about. If we just think the last bit first about different diseases, we are also interested in how diabetes, which is a metabolic disease, this isn't linked to mTOR, but just on that, how diabetes might affect COVID-19 development, because there has been links that diabetes, if you're diabetic, you're more likely to have an adverse outcome for COVID-19. We have been that at the start. We had the two projects going. We had mTOR inhibitors, and we also were looking at metformin and how maybe anti-diabetics could have an effect. But because we got this great data with mTOR, we moved forward with this. But I think what we'd really like to ask is if patients are on metformin, which also inhibits mitochondrial, which also inhibits the electron transport chain in the mitochondria, could that have a beneficial outcome in diabetic patients? So we have been working with some people who are not on here at UCLA, who are looking into patient records of COVID-19 to see if they can determine if how many had diabetes and how many had metformin or not. So I think that would be really interesting. mTOR and aging, mTOR inhibitors have been linked to increased longevity.
Mm-hmm. I think that's really fascinating.
That is, yeah. I don't know. It would be great to be able to get enough patient material to be able to separate them into different age groups or different if underlying conditions. At the moment, we've been incredibly lucky to get. We have 5 COVID-19 lungs at the moment. We've been lucky to get them. It's just more of an amount of material that's the stumbling block in that. But it would be great to maybe if we can move this forward into a clinical trial. Maybe most people who will be on it will be in the older cohort anyway, because they are probably more likely to be having severe COVID-19. So it would be interesting to be able to determine which patients might be more likely to respond to this mTOR inhibition than not. And it might be age, it might be underlying metabolic conditions. These are all questions we need to answer, yeah.
Mm-hmm. Or I was thinking, the fact that this inhibitor can also revert aging, like you were saying, that could also be aging-related inflammation that is at baseline level.
Yeah,
that could feed into this phenotype.
Yeah, because it is an anti-inflammatory, which is
so I mean, it does so many things. Anyways, it's so complex but fascinating.
So yeah, we would love to be able to answer the effects on the immune system. There has been actually a paper in Molecular Cell that came out a couple of months ago, which looked at glycolysis in infected macrophages, whereas we're more interested in infected lung cells, and they saw that there was an increase in glycolysis. I don't think they checked mTOR activity, but I wouldn't be surprised if mTOR activity was also altered, because it is known to have an effect in switching and in the immune cells as well into what type of immune cells are.
It's cool. Thank you.
Great. I don't know if there are any other questions. I'm not seeing any others. So with that, we'll just say thanks so much, Pete, for a really wonderful seminar. Really exciting stuff. I'm looking forward to seeing the follow-up studies. And thanks everybody. And then please join us next Friday for our next seminar. And if you have any questions about reagents or things that might be available at UCLA, please feel free to reach out to me and I can put you in touch with people. So thanks so much for joining everybody.
Thank you, everyone.
Great.
Thanks.
Thank you.
Bye.